All right, so I think our participant number is leveling off. So we'll go ahead and get started. Good afternoon, everyone, and welcome to the Palaeoverse Lecture Series. This lecture series is focused on software development and methodological approaches in paleontology, with the aim of promoting the use of new tools in the field and to give researchers a platform to showcase their work. With this series, we hope to shake up the usual type of lecture by having a mixed format, with time dedicated for the presentation, demonstration, and discussion of the software method presented. So typically these sessions will run about 50 minutes in total, and we'll run the series throughout the year on the last Thursday of every month. We have a really great lineup for the next few months, which we'll announce at the end of the seminar series, or today. And we're really looking forward to all of the upcoming talks. If you're interested in presenting a new method or software that you've been developing or implementing, please feel free to reach out to our organizing team. So before moving on, just a few introductions. So my name is Erin Dillon, and with me today, I have Lewis Jones and William Gearty. The three of us are on the development team of the Palaeoverse, which is an organization that aims to bring the paleobiology communities together to share resources, reach agreed standards, and improve reproducibility in paleobiological research. So if you want to learn more about the Palaeoverse, please check out our GitHub organization page, which will be in the chat, and our website. So that's all from me for now. Will now give you some helpful tips and instructions for this webinar, followed by Lewis, who will introduce our speaker. Thank you very much, and please enjoy the lecture. So over to you, Will.
Thank you, Erin. As Erin mentioned, the format of this seminar will consist of a talk by our speaker, followed by about 10 to 15 minutes at the end dedicated to questions and answers and discussion. My fellow hosts and I will help field your questions and moderate this discussion. Please note that this seminar will be recorded, but it will not be made available on our YouTube channel afterwards. We will stop the recording before the Q&A period in case you'd prefer not to be recorded. Before we get started, I'd like to quickly point out a couple of useful Zoom controls at the bottom of your screen. The Q&A button, you can use this to submit questions to our speaker. During the Q&A session, we'll then be able to read out your questions or invite you to turn on your microphone and ask the speaker directly. You can also use the raise hand button to raise your hand, and we can call on you and invite you to turn on your microphone. And again, you can ask your question live. There's also the chat. Currently, you can only chat with the hosts in case of technical difficulties, but we'll open this up to allow you to chat with other attendees, again, during the Q&A period. There's also closed captions, which you can turn on, and you'll be able to view the transcript at any time. I'd also like to highlight a couple of ground rules. Our aim is to cultivate an open, respectful, and inclusive environment during this lecture series where we can learn from each other. With this in mind, we ask that you please be respectful and kind when engaging with the speaker and your fellow attendees, particularly when asking questions and interacting with others in the chat. Any form of harassment or inappropriate behavior will be grounds for removal from the webinar platform. And for more information, please refer to the Palaeoverse Code of Conduct, which can be found on our website, and I'll put a link to that in the chat as well. With that, I'll pass it over to Lewis to introduce today's speaker.
Thanks, Will and Erin, for getting us all up to speed on how the lecture series will work. Today, I have the great honor of introducing Professor Daniele Silvestre. Daniele is a computational biologist and an assistant professor in the Department of Biology at the University of Fribourg, where he leads a research group focused on computational evolutionary and conservation biology. Within his group, Daniele develops computational methods and software to study evolutionary processes across different organisms, including plants, vertebrates, and invertebrate groups. His research group works on big questions, such as how does speciation, dispersal, and extinction shape biodiversity through time and space. Today, we're very excited to hear from Professor Daniele Silvestre on using neural network models to infer diversity trajectories and drivers of speciation and extinction. Please join me in welcoming Professor Daniele Silvestre. Over to you, Daniele.
All right, thank you very much for the introduction. Thanks everybody for organizing this symposium and this seminar series and for attending my talk. Today I would like to present a couple of models that we've been developing in my group. So most of the work is actually not made by me, but I'll be acknowledging the persons involved as we go along. Basically, yeah, our interest is in understanding biodiversity in deep time, how it evolves, how it changes over time, and how we can use the data available to make inferences about these trajectories and about the drivers of diversification through time. We developed a number of software looking at fossil data, molecular evidence, a combination of both, and trying to figure out, using probabilistic approaches and other types of methods, how to make the best out of these data. Today, we'll be focusing on fossil data and analysis of the fossil data based on neural network models. And so, before we start, I thought I would give you like a super brief version of what a neural network is. I'm not going to introduce the concept of fossils here because you probably know more than me about this, but neural networks maybe it's worth just getting a very short version of what they are so that we are on the same page when I mention the term. And so to do that, I'll go back to something really basic like a linear regression. And hopefully I'll try in the next 2 slides to explain what a neural network is in general terms and how it modifies a simple function like a linear regression. So I think we're all familiar with this type of equation, which is a linear function where we can, for example, predict the body size of a panda based on the amount of broccoli that they eat. And so to do this type of modeling, we are going to have a prediction, which is the body size of our pandas, a predictor, that is the diet or the broccoli that they eat. And this diet will be linked to a prediction through an effect size. And then we'll have an intercept, which is kind of a baseline body size of a panda that doesn't eat any broccoli. Okay, so this is how we write a linear function. We can extend this model to have multiple predictors. So if we don't only want to look at broccoli, but also genome and range size or whatever other traits, we can build a multiple linear regression model. And this is going to look like this. We can write it as an equation, so where we have an effect size for each predictor, and then we sum all of these predictors, all of these effect sizes. We still have an intercept, so the body mass of a panda under this model is predicted by the cumulative effects of these predictors through these effect sizes. So these are functions that we use all the time and that I think are familiar to most. They have some pros and cons. They are simple models, so they have parameters that can be directly interpreted. So we can easily take these effect sizes and understand what they mean, right? How they positively or negatively relate a prediction to a predictor. However, of course, these models are limited in the type of responses that we can generate through these models. So these models only allow for linear responses. So the body mass of a panda can only be a linear function of the broccoli they eat. And unless we specify this explicitly, there will be no interaction terms here. So here there is no interaction between, for example, diet and genome size. A neural network can be described in terms that are similar to these regression models. And in particular, it can be displayed like this. Here, what we're going to do is basically to introduce a hidden layer, so an extra layer in our model where we don't go directly from our predictors to our predicted values, but we have an intermediate step, one or more intermediate steps. And these are called hidden layers in a neural network. But the type of operations that we're going to do in a neural network are really similar to what we do in a regression model. So these are all weights, which are basically effect sizes. Only this time, we don't only have a single effect size that links diet to the next layer, but we have as many, in a fully connected network, we'll have as many effect sizes or weights as there are nodes in this hidden layer. So you see now that there are parameters connecting everything with everything. And this is important because it means that in a neural network, the response will be not just a function, a cumulative function of the effects, the individual effects of these predictors, but it will also incorporate interactions in there. So here we have the interaction part. Inside this hidden layer, we also have an activation function. And an activation function is typically a very simple function that transforms whatever numbers we put here into another set of numbers, but this transformation is non-linear. So it will be a simple function that kind of breaks the linearity. It could be some sigmoidal function or similar. And the purpose of this function is to break the assumption of linear responses. You see that this part of the network is actually very similar to what we had up here. So basically, we end up with a bunch of numbers that are a transformation of the initial predictors, and then we turn these numbers into our prediction. A neural network model has, again, pros and cons, so no model is perfect. The advantage of a neural network model is that it can allow nonlinear responses. And not only that, but you don't need to specify what type of function your response will follow. So it's not just either linear or exponential or something else. It can take virtually any shape. So we don't need to specify what type of function links the predictors to the outcome. It also accounts for interactions among all of the predictors. On the other hand, these parameters that are estimated are not directly interpretable. So unlike effect sizes in a regression model, we won't be able to directly interpret these parameters. And these models tend to be over-parameterized, meaning that they have way more parameters than they need. And this is a pro and a con at the same time. So we are going to need to take care of this risk of overfitting, basically, when using neural networks. All right, so this is the short version of what neural networks are. And so I will go straight into how we apply neural networks to understanding biological questions. And the first question is, can we use the fossil record to understand biodiversity dynamics in deep time? So people have been drawing curves of biodiversity trajectories for a very long time, and this has been extremely important to understand how clades evolved over time, how they replace one another, and so on. However, recent evidence showed that because the fossil record is inevitably plagued by non-random biases, so not only is the fossil record incomplete, but it's also biased in many ways. So some areas are sampled more than others, some are sampled more than others, some time beings are sampled more than others. So all of these biases essentially make the true biodiversity patterns unidentifiable under the standard approaches that have been used to draw these curves so far. And this has been shown in a couple of influential papers recently. And these papers were actually the ones that led us to developing a new approach that is based on neural networks. And the new approach is called Deep Dive. And it's using deep learning to perform diversity estimation using fossil records, using the fossil record. This is the work of Rebecca Cooper and collaborators, but mostly her PhD project that has now seen the light in this paper that just came out in Nature Communications. Here, the idea is that we can take the fossil record and describe the fossil record looking at a number of features that we can observe. So we can count, for example, in the fossil record, we can measure the sample diversity. That is a kind of one of the simplest metrics of diversity from the fossil record. Then we can take the range through diversity. We can count the number of occurrences, the number of singletons, and the number of sampling localities. And we can do this for each area. So if we have fossils, if we're aiming to estimate the diversity trajectory of a globally distributed clade, we can calculate these numbers for each time being in each area independently and individually. The idea is that then we can feed all these numbers that we wouldn't necessarily know how to directly link to a diversity prediction. We will feed these numbers into a deep learning model, so a deep neural network. And the output of this neural network will be an estimation of diversity at each time being, global diversity of the clade at each time being. So here we're going to use a special type of neural networks that is called recurrent neural network, where basically there is a certain interdependency between time beings, so that the model knows that time beings are actually a time series. And we try to take all of this data, all of these counts that we can observe in the fossil record and turn them into a prediction of diversity. Now, the problem is, how do we train, how do we optimize this model? Ideally, we would know for a number of data sets, both the fossil record and the true diversity, and use them to train the model, to optimize the model. However, this doesn't happen because for no clade do we know actually their true diversity trajectory. So what we do instead within the deep dive framework is to couple supervised learning with mechanistic simulations. So we use simulations to reproduce any number of artificial data sets where we create both the true underlying diversity trajectory and the fossil record. And the good thing of simulating data is that within these simulations, we can introduce everything, every bias that we know will affect the fossil record. So we can generate true diversity trajectories using a speciation and extinction process, but we will also simulate the geographic range of these species, for example, through a migration process. And we'll also simulate then the resulting fossil record. And here we will introduce all the biases that affect individual areas. They can be different across areas and through time. So we generate a number of data sets for which we know both the ground truth, which is the true diversity, and the incomplete fossil record. We can then use this data to train a deep neural network. And this neural network will take the features of the simulated data sets and turn them into an estimated diversity trajectory. We are going to need a lot of these data sets, usually tens of thousands or hundreds of thousands. But after that, we'll have a model that can take a fossil data set and turn it into a prediction. We showed with simulations that this model works under a range of different settings and that it outperforms state-of-the-art alternative methods. And this is particularly true. We have strong temporal biases or taxonomic biases or spatial biases. Once we have a trained model, we can take our empirical fossil record, feed it into the model, and get a prediction. Now, one thing we have to keep in mind when using supervised models is that there is a risk in how we extrapolate. So if our simulations that we use to train our model are different from the empirical data, then we have the risk of trying to extrapolate, trying to make predictions from this trained model that are outside of the observed range. So we need to make sure that our simulations are calibrated to the empirical data sets that we're actually feeding them to. And to do this, we compare the features of the simulated data sets with the features observed in our empirical data. And we need to make sure that the empirical data set fits within the range of simulated features. Okay, so the simulations have to be in the right ballpark to make sure that our estimates are reliable. Once that is also done, then we can apply these trained models to empirical data sets, as we did in our paper that you are welcome to check out if you're interested. Just an overview of how deep dive the software works. This is basically split into an R part that is coming up soon. There is actually going to be its own R package called Deep Diver that helps you basically preparing the input file for deep dive. So this will parse a fossil data set and create a config file with all the settings for the analysis. And then this will feed into a Python program that will do in parallel. So it will use multiple CPUs if available. It will run a bunch of simulations. It will adjust its own settings to make sure that the simulations are in the right ballpark, but you can also manually change these settings, obviously. Then once we have all these simulated data sets, we can feed them into a neural network and we'll run the training typically on a GPU if available, which will make it a lot faster. And after that, we'll get the predictions. So this is actually done in a pretty simple pipeline, all of these steps. So we start basically with a table with occurrence data. Each occurrence will have a mean, max, age that is basically the stratigraphic age of this occurrence, a locality information or longitude, latitude, some sort of spatial information anyway. This table is processed through our R package. In a few lines, we basically create the input file and a config file, and then these two input files feed into the Python program. I should mention that we can also calibrate our predictions to the true diversity that we observe today. So if the clade we are analyzing is living today, like mammals, then we can use the modern diversity to further calibrate our predictions, and this will feed directly into the model. Once we do this step, we create these two input files, the config and the deep dive formatted occurrence data, then we can feed them into our Python program. The Python program will be basically just taking this config file and run the rest of the pipeline. So we run the simulations, model training, and prediction. And as an output, we'll be able to observe how the training worked. We'll have some measures of model accuracy based on a test set, and we get our empirical predictions. This is how we can use deep learning, supervised deep learning, to obtain estimates of diversity through time. As we saw for the elephants, and as you can see in the paper that just came out, like we predicted for the elephants, a massive drop in diversity in the recent, but also a number of other fluctuations in diversity through time. And so the next question would be like, what is driving these diversity changes? So now we have this deep learning model, which we can use to estimate these diversity changes, but can we say something about the causes of these diversity changes? And this brings me to the second model that I wanted to present to you today. That is based on a different analysis of the fossil record, using Bayesian modeling, but still using a neural network. And so this is based on, so the idea here is that we want to basically try to use possible predictors as and understand their importance in shaping this curve. And we are going to do this through a birth death process. So this builds upon a program that we've been developing for several years. This is not based on neural networks in its default version, but is instead a Bayesian program that takes fossil occurrence data and jointly estimates their true duration, their true longevity through a preservation process. So it will estimate sampling or preservation rates through time. And as part of the estimation, it will also get estimates of speciation and extinction rates that can, for example, change over time. This is implemented in a software called Pyrate that has been around for a few years already. And within this framework, so this is not using deep learning, it's a standard probabilistic approach using Bayesian inference. And we have a number of flavors of the birth-death process that allow us to model the speciation and extinction rates, which ultimately determine diversity trajectories through time, as a function of different things. So we have a number of flavors of birth-death models that we can use to test different hypotheses. For example, we have constant rate models that can be compared against time variable models, where these rates vary over time. We can make rates be a function of a discrete rate. So, for example, extinction rates can be modulated by the presence or absence of a particular trait. We have models where we can try to introduce multiple variables as predictors using a multiple linear regression model, as the one that I mentioned earlier, and a number of other things. Now, the problem with these methods is that you need to specify what you want to correlate with speciation and extinction rates, and you need to define what function is going to be your response variable, right? So a response function. So if you want to fit a model where you have where you want to regress, for example, temperature against speciation rates, you need to decide if this relationship will be linear or exponential, for instance. And it will be difficult to make, for example, temperature interact with another trait. So as a solution to these, and with the aim to try to put together all of these different flavors of birth-death models within a single framework, we developed the BDNN model, which is a birth-death neural network model, where essentially we express the speciation and extinction for each species and for each time being as a function of a number of traits, time itself, phylogenetic relatedness, whatever we want, basically. So we can use a number of predictors here that we think are relevant. And these predictors will feed into a neural network. And the output of this neural network will be a species-specific, time-specific speciation rate or extinction rate. Because we are using a neural network, we don't make any assumptions about the specific shape of these correlations, and we can account for interactions between all of these things. As this is a very flexible model, we can use time series as input. We can use time itself, so the speciation and extinction rates can vary as a function of time itself. We can use categorical trace and continuous trace. This model will feed directly into the same framework that we've seen before. So before, in a standard pyrite model, we'll have the fossil record of which we calculate the likelihood based on a preservation process and a speciation extinction process. Here, we basically replace this birth-death process with a birth-death process where the rates are modulated by a neural network. Now, one thing that I did mention earlier about neural networks is that they tend to overfit. They are over-parameterized, right? There are so many parameters here, many of which are likely redundant. And so the risk is overfitting. When we do supervised learning, there are some rules in how we optimize the model to prevent overfitting. But this model is not supervised. We don't simulate anything. We take the empirical data and we analyze it using a probabilistic framework, this Bayesian framework. So we need to make sure that our neural network model doesn't overfit the data. Otherwise, we risk getting spurious outputs. And so overfitting is something that is very, so it's a risk that is very well known in statistics, and there are a number of measures that we can take to prevent overfitting. One of them is using priors on our weights. So we have normally distributed priors on all of these coefficients. These normal distributions are centered in 0. So we favor a priori a model where these effects are basically not doing anything. So where there is, we favor a null model where the rates are constant by our a priori assumptions. This is a typical feature of Bayesian neural networks. But on top of that, we add another way to do model regularization, which is basically a way to prevent overfitting. And this is by introducing another regularizing parameter that is essentially raising these species time-specific rates to the power of a number that goes between 0 and 1. And so this is a single parameter that performs what is called Bayesian shrinkage, which is shrinking the parameters around their mean unless there is evidence that they shouldn't be. So if this parameter is set to 1, then the output of our network will be unchanged, and it could be a rate that changes over time like this. But if this parameter is estimated to be close to 0, then our resulting rates will be kind of shrunk around their mean. And this is very important because essentially, if we run this analysis on data sets where there is no rate variation, despite the fact that we have all of these parameters, we will estimate this parameter to be really close to 0, and our resulting rates will be essentially constant. So we regularize the model and avoid overfitting. This model was tested again against a number of simulations. The main developer of this model is Thorsten Haufer, who has done most of the development and is the first author of a paper that is coming up soon, very soon, hopefully. And we analyzed our elephant data set using this model. And so here we use a number of predictors for these elephant data sets, and we had climate variables, biogeography, we had time itself, we had phylogenetic relatedness as one of the predictors, and we introduced humans overlap, spatiotemporal overlap with humans as one of the predictors. So we fitted this model on our proboscidean data set, and what we are able to get from this model is species-specific rates of speciation and extinction. These are some examples. We get these distributions. So these are posterior estimates of speciation and extinction rates for each species. We can marginalize these rates through time to get average speciation rates and extinction rates through time. But then we can also use tools from explainable AI, which is a branch of AI research trying to make sense of which predictors are driving the change in speciation and extinction rates. So we can use a number of tools from explainable AI to rank the models and quantify, to rank the predictors and quantify which predictors are most important to determine changes in speciation and extinction rates. So there is a number of metrics that we can use to do that, and we can use them to rank the predictors. And what we found is that for speciation, there are ecomorphological traits that are very important in shaping them. Time itself is one of the parameters explaining changes in speciation rate, and so is biogeography. For extinction, we found that by far humans are the most, overlap with humans is the most, single most important factor affecting extinction rates in past extinction rates in proboscideans, but also with effects with biogeography and ecomorphological traits. And we can then use partial dependence plots to actually measure the effect of these predictors. So for example, here we were able to plot how time in interaction with an ecomorphological trait affects speciation rate. All of these dots are species with their the time where they lived and their trait, and you can see how the rate changes or the estimated rate changes as a function of these two things. You see here we're not assuming a linear response. In fact, we are even estimating an interaction term between these two variables. We also see that there are variations linked to biogeography, with speciation rates on islands being significantly higher than on continents. For extinctions, we found that the overlap with modern humans is linked with that 15-20-fold increase in extinction rates compared to the baseline, compared to a species that did not overlap with humans. And this is while accounting for everything else, right? So this is partial dependencies, meaning that this is isolating the effect of humans, and in this case, an ecomorphological trait as well. We also find a higher extinction rate linked to living on islands, which is not exactly unexpected. And we found in this case that there was a minor effect of regional climate change on extinction of proboscideans. So in this case, we were able to pinpoint the massive drop in elephant biodiversity in the recent, basically pinpointing to the overlap with humans as the single most important cause for this extinction. The BDNN model is implemented in Pirate, is available online. The paper is coming up soon, but the tutorials that Thorsten prepared are already out there. They're very detailed, very informative, and so you can follow a number of a few simple steps to actually set up this BDNN analysis. All you're going to need is a fossil occurrence data set, like you would use for other Pirate analysis. So basically, fossil occurrence is assigned to a taxon. And then you will need a trait table, which is basically a CSV file with species names and a number of traits that can be discrete or continuous. You can also include time series in the analysis, and they will be taken into account within the BDNN model. If you want to know more about these models, you are obviously very welcome to contact us. Go and check the online resources, but also to join our workshop that we plan adjacent to the GSA meeting in September. So we'll be demonstrating basically these models and others in a one-day workshop. And so we hope many people will be able to join. With this, I would like to thank again all the members of my group for contributing in different parts of this work and for being wonderful collaborators. And I would like to thank you all for listening. Thank you very much.
Thank you, Daniele. That was amazing. so much covered. Yeah, so we've got a few immediate questions here.